first want to apologize to you because my voice is a little scratchy. It seems that I've had something going on. I don't know if it's a mild cold or some laryngitis, but anyway, with that said, just bear with me. I'll try to make this one short and sweet so you don't have to put up with this scratchy voice for too long a time. I'm going to be talking to you today about the cardiac conduction system, which is the mechanism that keeps the heart beating, and then briefly at the end talk just a little bit about how the nervous system controls this as well. So let's get started. All right, by now you have seen hopefully a slide or at least pictures of cardiac cells, cardiac muscle cells, and how those cells are locked together or joined by structures that are called intercalated discs. Okay, so an intercalated disc consists of a couple of things that are really important. One, and this picture you can sort of get an idea of what I'm talking about. It's not the best picture, but it will work. But you have desmosomes, which are junctions between the cells that hold the cells together somewhat like little spot welds. So they're very, very tightly held areas between the cells that keep the cells from pulling apart, because think about what the heart does. The heart is constantly beating. Those cells are constantly contracting, so they would be pulling against each other. So these spot welds, or these desmosomes, weave together sort of like sheets of cardboard in a box to hold those cells together. But the other important function, or the other important structure between cardiac cells, are gap junctions. Now, gap junctions are little channel-like structures between cells, and they allow for things to flow from one cell to another. Now, that's going to be really important as we continue talking about, talking about cardiac conduction, because those gap junctions allow for signals, electrical signals known as impulses, to transfer from one cell to another by the transfer of ions, charged particles. You know what ions are. By allowing those charged particles, those ions, to move from one cell to the other. So it allows for communication between the cardiac cells. Now, these cells within the heart are stimulated by nerve fibers that we'll talk about in a few minutes, but also heart muscle is self-excitable. So it exhibits automaticity, automatically stimulates itself. So it's also called autorhythmicity, automatically rhythmic. So they have a certain rhythm that is inherent or intrinsic to these cells, and when this happens, it allows the heart to conduct as a unit. So you don't have one cell contracting and then stimulating another cell to contract, and another cell to contract, and another cell to contract in a long, drawn-out process, similar to the way a nerve cell would stimulate a muscle. But because of those gap junctions allowing the flow of ions, you have a very fast transfer of that electrical conduction or that impulse from one cell to another, so the heart does contract as a unit. Okay, let's see what else I wanted to tell you. Now, remember what causes an impulse. You remember the action potential from 201. Remember, the action potential is that impulse that travels down the cell and causes something to happen. We started with the motor neuron, remember, and we sent an action potential down the motor neuron, and then the release of acetylcholine stimulated the muscle cell to contract. So when you hear me talking about impulses, think back to what an impulse actually is. It's that transmission of the charge, or transmission of the electricity, the change in potential that's going to lead to an action. So you'll probably hear me simply call these impulses as we're talking about these. Now, let's go on and move to the next slide. And I'm going to skip to some information in a few minutes, or I'm going to skip to a picture and let you refer to the picture and this slide as well. So when you're making your notes, you may want to move back and forth between the two a little bit. But now remember, you've got heart cells, the muscle cells, that are going to specialize and contract. Their job is to contract to keep the heart beating. But there are other cells within the heart that don't contract. They're specialized to initiate that that rhythm. So they initiate and distribute that impulse throughout the heart. So these cells are said to be autorhythmic. They generate their own impulse, and then they depolarize continuously, and by doing so, they send that impulse or that action potential all throughout the heart. So these cells are self-stimulated, and they're responsible for keeping the heart muscle contracting, and it serves as sort of an in-house conduction system that allows the heart to keep beating even without the input of the nervous system. Now remember, when we looked at muscle cell contracting, skeletal muscle cell contracting, you always had that motor neuron stimulating those cells to contract. Well, if you've watched Discovery TV or Health TV or even food television with the really strange bald guy that eats weird foods all the time, I can't think of his name. But anyway, you may have seen a heart beating outside the body. The bald guy I was talking about eats beating cobra hearts. And of course, those would have to be outside the body for it to be safe to eat them. I don't know that I'd do that anyway. But think about that. That heart is still beating outside the body, whether it's the cobra heart or a human heart in a transplant situation. If that heart is still beating, there are no nerves attached stimulating that heart to beat. So those cells that are specialized to keep the heart beating are depolarizing themselves, and therefore keeping that in-house conduction system working. Now that's not going to continue for an extended period of time, but for that short period of time that you would take the heart out of the cobra to eat it, or from one patient into another, that short period of time, the heart would continue to beat. Okay, so let's see, where did I want to go next with this? Now, we'll move on to the picture that I wanted to show you, but the information on the previous slide is going to tie in with this picture. But I think it'll be easier for you to understand what I'm talking about if you see the picture and can sort of follow the pathway as we talk about this. So we're still talking about these cells that automatically send out these impulses and keep the heart beating. So these structures comprise the cardiac conduction system. Now these autorhythmic cells are cells that don't contract, and they're found in different places located throughout the heart. They specialize to initiate and distribute those impulses throughout the heart. So let's begin this pathway. You should be familiar enough with the heart anatomy by now that some of this should look really familiar to you. But we'll start right here with number 1, the SA node. This is short for sinoatrial node. This is also known as the pacemaker of the heart because it's responsible for starting these impulses and keeping the heart beating. Okay, so the SA node is located in the right atrial wall. So here's our right atrium. And it's located just right beneath the opening of the superior vena cava. So the cells within the sinoatrial node are going to initiate one impulse after another at a rate of approximately 75 impulses per minute. Now when this happens, it's going to send impulses throughout the atria, okay, traveling from the right atrium through all those gap junctions in the cells over to the left atrium, down the walls of the right atrium, and down to another structure that we'll get to in just a minute. So all of this depolarization is spreading through these gap junctions, through the atria, and the atrial myocardium is then going to be stimulated to contract. Now, as it does so, it's going to send blood through these AV valves. What's AV? Atrioventricular, exactly. You knew that already, I know. All right, so the blood, once the atria contract, the blood is going to be forced through the tricuspid and the bicuspid, let's do this way, the tricuspid and the bicuspid valves, and then the blood, of course, is going to go down into the left and right ventricles. Now, as that's happening, we've got more going on. Okay, so we were talking about that impulse being transferred. So it's going all through the walls of the right and the left atria, and also down to another structure here known as the atrioventricular node. Atrioventricular node. That should tell you that it somehow is going to be involved in both the atria and the ventricles. Now, I want to tell you what's going on here, but I want to sidestep for just a minute as well. Between the atria and the ventricles, there is a fibrous skeleton that is composed of connective tissue, and what it does is to surround the valves and provide like a floor to the atria. It also serves as a point for cardiac muscle to attach. But because it's fibrous connective tissue, it does not conduct impulses. So you don't have the gap junctions and those desmosomes between the atria and the ventricles there. So the only way that that impulse is going to pass is going to be through the structures that we're going to talk about. So if the impulse is going to pass from the atria to the ventricles, it has to go through these autorhythmic cells. Okay, so this wave of depolarization now is going to reach the atrioventricular node. Now, once it reaches that when it reaches this atrioventricular node, then it's going to pause. This is an area where the stimulus or the action potential pauses for just a minute to allow the atria to completely contract before that signal is sent down to the ventricles. Okay, so from the AV node, now remember this is the only pathway from the atria to the ventricles. From the AV node, the stimulus is going to pass through the atrioventricular bundle, okay, number 3 on our pathway. Now, as this is happening, you've got this interventricular septum, which is myocardium between the ventricles, right? Separates the two chambers. So we're going down through the atrioventricular bundle, and then it splits into left and right AV or atrioventricular branches. Let me back up just a second. I didn't point out this term on number 3. The AV bundle is also known as the bundle of His. You may hear that from some of your instructors. You may also see that in your text, but it is just another name for this. Now, as this wave of depolarization is continuing down these bundle branches, left and right bundle branches, about halfway down the interventricular septum, these become Purkinje fibers. Purkinje fibers are going to go all the way out and stimulate the walls of the ventricles, okay, as well as the interventricular septum and the papillary muscles that we have that are shown here as well. Okay, so what happens as this impulse travels down toward the apex of the heart, these cells are being stimulated. So you really start seeing contraction from the apex up. All right, so when the apex begins to become depolarized, or the cells in the apex begin to become depolarized, they're going to start contracting, and you have a squishing of all of the myocardium, and of course that's going to force the blood out through the aortic valve and the pulmonary valve. All right, let's see, what else did I want to tell you about this? I think that's pretty much all I wanted to tell you on this particular picture. But I do want you to pay attention to this pathway. It's a very predictable pathway, and maybe predictable is not the word that I should use, but it's a very easy to learn pathway because it always travels in the same direction. Your signal is going to start at your sinoatrial node and then depolarize the cells of the atria. The atria are going to contract and force the blood through the valves down into the ventricles. Now, while this is going on, that stimulus is being sent to the atrioventricular node, which is going to pause for just a minute to allow the atria to complete their contraction, and then the stimulus is going to travel through the atrioventricular bundle, the bundle branches, and then the Purkinje fibers. Now, one thing I do want to point out here before I move on to the next slide is that these fibers, these Purkinje fibers and the stimulating part of these, the cells that are actually conducting this impulse, don't reach out and touch every cell of the myocardium. And this is where those gap junctions play a big role, because not every cell is touched by one of these stimulating fibers or one of these conducting cells. Those gap junctions allow for the cells that are touched by a part of this conduction system to pass that electric current through the flow of ions into the adjacent cells, so that all of the heart does depolarize and contract as it should as a functional unit. Okay, let's move on. Now, when you see a video that should be coming up soon after this one, you're going to get some information about an ECG. Okay, you may also hear this EKG. The K is from the German. I'm sorry, I believe it's German. I'm not 100% sure now that I've said that. But it is an electrocardiograph. And this is actually showing in a nice organized manner what's going on with this depolarization. So you can look at these ECGs and see what a normal heart rate, a normal impulse conduction should look like, and be able to use what you know is normal to diagnose something that's not going on as it should. So when let's change colors here since this one is blue, we'll go with red. When the atria are depolarizing, now remember we're starting here with the sinoatrial node, sending that signal down to the atrioventricular node. So when the atria are depolarizing, we have a very characteristic P wave that is created. Then we have the ventricles depolarizing, which gives us a QRS complex. And then when the ventricles repolarize, or coming back to their normal state, we have a T wave that's created. Now you're going to hear much more about this, but I wanted you to just look at this picture so you can see what's going on by using those electrodes and putting them on the patient's body. Those electrical stimulus, or the electrical stimulus and the pathway can be picked up. So when you have stimulation in a particular part of the heart, then a particular wave will be created on these ECGs. And you're going to get much more than that from the paramedic guys. We hope are going to come over and do a video for you, and I know they'll do a wonderful job on that, so I look forward to you seeing that. Okay, let's move on with one more slide and wrap this up. I know you're tired of hearing my scratchy, squeaky voice. Now, remember, the heart has an intrinsic conduction system that keeps it beating. But the heart can be affected by the autonomic nervous system. So the autonomic nervous system can modify the timing of those impulses that are sent throughout the cardiac conduction system. Let me go ahead and change colors here because it looks like this one's not going to work. Let's go with the green. Okay, so within the brain, we have cardiac centers that are inhibitory and stimulatory. Now, these are located within the medulla oblongata. Remember that from your brain anatomy? You better. Where's it at? Located in the brain stem, exactly. So you've got a lot of vital functioning centers within the medulla, and the cardiac centers are two of those, or one of those that has two different functions. So these centers receive information via receptors and then cause a reaction to occur. So if we just look at examples, these carotid baroreceptors, baro equals pressure. So these receptors in the carotid arteries and located in other areas as well are going to pick up changes in pressure within these vessels. So let's say these baroreceptors detect an increase in blood pressure. They're going to signal the parasympathetic nervous system. The parasympathetic nervous system is a part of the parasympathetic nervous system is the cardioinhibitory center. So it's going to send motor impulses driven by acetylcholine, our old friend there. Remember in muscle contraction? Well, this is a different action here for the acetylcholine. It's going to slow the heart rate. So by slowing the heart rate, it is in effect going to decrease the blood pressure. So the parasympathetic fibers are the brakes of of the cardiac conduction system. So when the parasympathetic nervous system is stimulated, then the heart is stimulated to slow that impulse conduction. So it does so by changing the way that the cells are depolarized. Now, on the other side of that, when the when the sympathetic nervous system is activated, it's like releasing that brake, taking that brake off and pressing the accelerator. So the sympathetic nervous system is the cardioacceleratory center. And these sympathetic nerve fibers are going to stimulate the pacemaker or the sinoatrial node to fire more rapidly, and by doing so, that is in effect going to increase the heart rate. Of course, this is going to happen during times of stress, anxiety, excitement, exercise. During all of these times, of course, the heart needs to pump faster. The heart pumping faster gets more nutrients, more fuel, more oxygen out to the body. So it's important during these times that the cardioacceleratory center, or the accelerator portion of this system, is stimulated. Okay, now your instructors will probably go a little bit deeper on this in class if they choose to, but I wanted to give you some basic information on this and get you started. So read your text as always, listen to your instructors, and keep watching the videos, and hopefully next time I talk to you I won't sound quite so scratchy. So thank you for putting up with me today, and I'll be back and talk to you again soon. Take care.